This is our modern marvel of the day. It takes sunlight, dirt, and air, then uses physics to turn rock into breathable air. Pretty great, but there's something odd about it. Let's zoom in on the star ingredient, this humble little enzyme called Rubisco. Now this thing is amazing, but it's also notoriously klutzy. Its job is to grab carbon dioxide from the air and get the whole process going. Except every now and then, thanks to a quirk of chemistry and physics, it grabs the wrong thing. And when it does, well, everything changes. Hi, and welcome to this channel. Today we're going to see how life copes with a fundamental flaw in the machine that makes food. And along the way, find out what breakfast has to do with breathing. It all starts here. Every apple, every loaf of bread, every bowl of rice began as air. Plants use sunlight to pull carbon dioxide from the atmosphere, split water for hydrogen, and stitch the pieces into sugar. That's photosynthesis. And Rubisco is the star of the show. But let's put the whole play in one picture. First, light reactions inside these guys called thylakoids capture sunlight and turn it into ATP and NADPH. Think of those as energy and reducing power. Then the Calvin cycle takes that power and uses it to bolt carbon dioxide onto this 5-carbon sugar and build carbohydrates. At the gate to that process sits Rubisco. It scans the molecular landscape for a small out-of-place carbon dioxide, finds one, it grabs it and drops it onto the sugar. And only then does the whole wonderful cycle start to turn out food. And when carbon dioxide is plentiful, the cycle hums along. Triose phosphates leave as sugar, and the plant stocks the pantry. But when carbon dioxide falls, the system stalls. That's when Rubisco's bad habit shows up. See, Rubisco can't always tell carbon dioxide from oxygen. On hot, dry days, plants close their stomata to save water. Inside their leaves, carbon dioxide levels drop, and oxygen becomes the wrong key that still fits the lock. Rubisco grabs it and tries to start the Calvin cycle. No surprise, that doesn't work. What happens next depends on the plant, but it always involves a lot of backtracking. In some, an enzyme chops the 5-carbon sugar back down to 3 carbons, and another enzyme recycles those to make a new 5-carbon sugar. In others, the cycle keeps churning even without carbon dioxide. But either way, the plant wastes energy and reducing power and even releases carbon dioxide, undoing some of the work you just did. Left unchecked on hot, bright days, photorespiration can chew a serious dent in yield. So plants have evolved several ways to hack around Rubisco's flaw. The most widespread, C4 and CAM, take carbon dioxide out of the air and concentrate it near Rubisco. One way to do that is with a front-end pump. Some plants have evolved to grab carbon dioxide in one part of the leaf and shuttle it to another. This is C4. Here's how it works. In the Mesophyll cells, the enzyme Carboxylase grabs carbon dioxide and turns it into a 4-carbon acid. That acid moves into nearby bundle-sheath cells, releases carbon dioxide, and pushes oxygen out of the conversation. Rubisco sits in a carbon dioxide cloud, making the right choice easy. It costs a few extra ATP, but under high light and heat, that's a bargain. These plants, which include maize, sugarcane, and sorghum, thrive in the tropics and warm summers. Think of the commute between 2 cities. If the traffic is terrible, you might take the train. You won't get there as fast, but you'll arrive and the trip will be less stressful. C4 is kind of like that. On the downside, that pump costs ATP, so in cool, dim conditions, the energy bill can outweigh the benefit, and it needs the right leaf plumbing to work. So C4 really shines in bright, hot fields. Another option is to move steps to night and day. This is CAM. These plants open stomata at night when it's cool and dry, fix carbon dioxide into malic acid, and store it in vacuoles. Daylight closes stomata to save water. The acid is decarboxylated to feed carbon dioxide to Rubisco behind closed doors. Perfect for desserts and epiphytes like cacti, succulents, and pineapple. CAM plants grow slower than C3, but they're excellent at conserving water. Think of CAM as a 2-tank system. The night tank fills with malate while the air is kind. The day tank feeds the Calvin cycle while stomata stay shut. You trade speed for survival, and in harsh climates, that's a winning trade. And finally, some tiny organisms build microrooms for Rubisco. Algae and cyanobacteria construct protein shells called carboxylases. Other types of microcompartments called pyrenoids form from liquid-liquid phase separation. They pump in bicarbonate, then convert it to carbon dioxide inside the compartment, raising local CO₂ where it counts. Each of these hacks comes with trade-offs. For instance, why not just make Rubisco better? Faster Rubisco tends to make more oxygen mistakes. And Rubisco is huge, slow to assemble and fussy about helpers. Evolution found it easier to work around the flaw pumps, time shifts, microrooms, than to reinvent the lock and key. For the same reason, engineers today are taking multiple approaches to boosting plants' carbon capture. One frontier is to import ready-made carboxylases or pyrenoid-like condensates into crops. Give Rubisco a private carbon cloud without re-engineering the whole leaf. Early prototypes show targeted shells and CO₂ handling enzymes can form in plant cells, but it's still work in progress. Wiring up the details. Another path is to shorten the detour of photorespiration with custom enzymes. By rerouting molecules through new pathways, plants can save energy and carbon that would have been wasted, especially on hot days. Less backtracking, more growth. Yet another frontier is to add C4 traits to a C3 crop. That means new enzymes and new leaf plumbing, a version of Kranz anatomy and cell-specific expression. It's dozens of genes and careful choreography. But pilot plants are inching toward the blueprint. Whatever the approach, gains come from stacks. Genetics, water, soil, pests, and management all work together. No single player can save the day, just like no single ingredient could turn sunlight into breakfast. Take a bite of your favorite breakfast food. Now imagine making that dish without the physics that makes breathing possible. Yeah, that's right. There's nothing in there that could have existed before Earth's atmosphere had enough oxygen to support animals. You can get pretty far using only the carbon, hydrogen, and oxygen that are in plain old water. Air miners, that's what plants are. They turn a thin gas and a hard rock into breakfast. And when their tools break, they hack around the flaws. Sometimes that means concentrating more carbon near the site of the breakdown. Sometimes it means changing when and where the steps take place. Sometimes it means building a tiny room for the assembly to happen, and life finds a way to go on after a catastrophic evolutionary accident. And it's a good thing too, because if there were no flaws in our molecular tools, well, we'd never have gotten breakfast in the first place. Thanks for watching this episode.